Okay, we can start. So welcome everybody. Welcome to the third day of the Quantitative Systems Biology School 2021. So today we have Antonio Celani from the International Center for Theoretical Physics. So Antonio is a physicist. He did his PhD in physics from Politecnico di Torino. After a couple of postdocs, he was a CNRS research fellow in France. And he was also the director of the Institut Pasteur as a research director. And there, most of his work was really on statistical fluid dynamics, stochastic control, and thermodynamics. And now, from the past few years, after moving to ICTP in particular, his interests have shifted more towards machine navigation, foraging, and in particular using machine learning techniques to explore those areas. So he'll give 2 lectures on olfactory mechanisms. So today he'll start off with an introduction, olfactory search and navigation. So Antonio, to you.
Thank you, Vijay. Let me start by outlining the plan for these 2 talks. So today it will be mostly an introduction to the problem of olfactory search and navigation based on a few selected examples. There will be basically no formula, no mathematics, mostly about exploring selected experiments and try to motivate the interest in studying olfactory navigation and search. Today's lecture will also be separated in 2 parts. So in the first part, I will be focusing on examples based on insects, and it will be about how insects locate as a source of an odor, or what do we know about it from the experimental level. And the second part will be about how homing pigeons, so an entirely different kind of animal, deals with navigation on much larger length scales and time scales. Tomorrow, we will totally change gears, and so tomorrow will be a talk with a high content in mathematical language, where I will introduce and describe the basic concepts and ideas behind the theory of decision processes with full information and with partial information, which will turn out to be a useful language to address problems like olfactory navigation, as well as many others. Okay, so let's get this started. So our story for today starts essentially one century ago. So the person here in this picture is Henri Fabre, who was an entomologist who has left us with a wealth of information on the habits and the behaviors of several insects in many books. And we will take our start from one particular page of his books, which are called Souvenirs entomologiques. And in his beautiful prose, Henri Fabre tells that starts telling about one night that he cannot forget, and that he is going to call the night of the giant peacock moth, which is the moth that you can see here. One morning, on the 6th of May, he can see that there is a female moth that leaves the cocoon, so completes the metamorphosis from caterpillar into butterfly, and immediately he takes this female moth and puts it under a cover, a metallic cover, and just because he wants to observe its behavior later on. And he says that I was in fact very, very lucky, because the same day, around 9 in the evening, when all my family was going to sleep, there is a big commotion in the room next to mine, and the small Paul, who is Fabre's son, goes back and forth and jumps and just like crazy. And then Paul addresses his father, says, "Come here, come here, come here. See that there are butterflies as big as birds, and the room is full of them." And then they enter this room with a candle in their hand, and what they see is an unforgettable view. So with a soft flapping of the wings, these big butterflies fly around the cover which contains the female. How many of them? They come from all points, and I don't know how they've been alerted to the presence of the female, but in fact they are here, 40 lovers who hurry to pay their tribute to the newborn female which was born that morning in the mystery of his own cabinet. So Fabre at the time already formulates an hypothesis that there might be some scents which are analog to what we call odors, such scents which have which are very faint and probably totally imperceptible for us, but still they could attract the males coming from a distance. Then, if you read the following pages, then Fabre goes on and explaining what are possible hypotheses for this attraction, long-range attraction of male moths from female moths, and it's really an entertaining read, and I advise all of you to take a read of these beautiful pages. Okay, so what is olfactory communication in general? Well, it's a communication. It's a communication between which can take place between animals, such as in moth, from the female to the male, or it can take place between plants or other animals, not the same species, which convey some message willingly or unwillingly, and this message is actually, as any message, goes through a series of steps, which can be described in a very abstract way, as starting from a message which is sent out by the sender, and it's transferred towards the receiver in a way that involves the presence of a channel. And for olfactory communication could be short range or long range, but typically what we are interested in is situations in which the channel is the air, the atmospheric air. That's typical for an odor. It can also take place in water, but we will not discuss this particular examples, even though all the kind of high-level discussion that I will be making are also relevant for olfactory communication in water. So when the signal arrives at the receiver's end, then this is what is now we can call a stimulus. Is then carried on from the peripheral to the central nervous system, and this then results in some behavior. So, of course, all the neuroscience takes place in this part which I depicted in yellow, which, however, I will not be discussing basically at all, or just carefully avoiding, in fact, this part. You will hear lectures from my colleagues who are absolutely experts in the field, and so you will be able to patch up the things. So what I will be mostly focusing on is first on the peculiarities of how the message is sent and transferred, because this involves a part of physics in chemistry also, but how the message can be delivered from the sender to the receiver, and the fact that the channel is atmospheric air and it has to undergo the constraints of the physical medium, which is there. It's very important because it shapes the signal in such a way that actually the message is very much dependent on what happens along the channel. So if you wish, the channel is part of the message. And then I will just jump, considering basically all the very relevant part, which is how the signal is actually received, relayed, and analyzed by the brain. Consider this a little bit as a black box and observe what behavior ensues from this chain of signals that are observed by the receiver. This way also is instrumental to the fact that eventually we will like to describe this process in an algorithmic way, and in this sense, what which takes the place of the brain here in this part could be replaced by any other kind of algorithm which processes information and turns it into decisions. Okay, so olfactory communication is very special in its own, and when we compare it to other forms of sensory communication, it stands out for some specific character which are quite important. Let's, for instance, compare it with the visual communication. So what happens if you can see an object? So we will be referring often to a problem of search. So let's compare visual search with chemical search. So visual search works on a medium range, okay? Because if the distance is too large, we just cannot see the object. Whereas chemical communication can work on much longer distances, as we will see. The transmission rate of a visual signal is extremely fast. It's just essentially the speed of light. Whereas the chemical signal is generally low and depends on the property of the channel communication that is the atmosphere. An advantage of chemical communication is that it can flow around barriers, whereas of course vision is impeded by obstacles. But the striking difference between chemical and visual communication is that once you receive the signal, to decode the position of the sender from the chemical signal is by far and large a difficult task as compared to what you can do if you can see your target. There are basically two extreme conditions in which the channel, that is the atmospheric air, can work. If the air is still, the only way for other molecules to travel from the sender to the receiver is to just diffusing through air. But diffusion in air is a very slow process, and it would take an enormous amount of time, several hours, just to make a few meters. Of course, this is basically never happens that the air is perfectly still. Air typically has wind, and wind can carry a signal faster. But the drawback of having wind is that the flow becomes very quickly unstable, and therefore it becomes turbulent. And turbulence, as a consequence, that the signal gets distorted and mangled, the odor gets mixed with other odors, with the clean air, and this is what makes the system difficult. So there is a trade-off. If you want your olfactory system to travel fast, you have to deal with the fact that it will be subject to a strong nonlinear perturbation in the original form of the signal. Okay, so in summary, what happens for moths is the following. Let's recap very briefly what we know. So moths live more. So I'm talking about not all moths, just a significant number of families of moths which have this lifestyle. They live most of their life as caterpillars. Often they are pests, so they ravage crops. Then they undergo metamorphosis and they turn into moths, into flying moths. And then they typically live just a few days in this state of moths. And in these days, basically, their only objective is mating. Very often, these moths do not even have functioning mouth parts, so they just live on the lipids that they have collected during their life as caterpillar, and they just use them out until they die. They die out in a few days, and every day, basically, in a given range of hours of the day, typically at dusk or during the night, depending on the species, they set ourselves into this mating exercise, which consists in the females start emitting tiny amounts of these specific odors, which are called pheromones. So they have a gland, and they emit a very, very minuscule amounts. We're talking about nanograms per hour of these compounds, which are highly volatile. And they're typically females sit on a tree, and they don't try to move around to make any kind of rendezvous with the males. But they sit there, and they wait for males to approach them. And it's been shown in the literature quite consistently that if you release males at a distance of hundreds of meters away from the female, they have a very high probability of getting to the female. So there are anecdotal situations which it's told that several kilometers distance could also so some male moths could be able to reach the female at kilometers of distance, but this is not extremely well quantified. So when the female starts emitting a signal, which is just a continuous or sometimes sparse emission of these pheromones at this very low concentration, this is a very specific message sent to the males of its own species. So it's a particular blend of odors with a specific concentrations. However, these signals, like I told you before, as soon as it gets caught by the wind and transported downwind, gets destroyed basically by turbulence in a sense, and because they're like I said, there's essentially clean air which erases part of the messages, and therefore when the male receives this message, it is extremely distorted with respect to the initial distribution of concentration close to the female. But nevertheless, males are able to decode this message, and therefore take action and start flying. And in a process of continuous search, moving around and collecting odor concentrations along the way, they can fly upwind and reach the female. Okay, so this is basically the very qualitative description of what happens. What matters for the male is are at least the three characteristics of the signal. One is the intensity, of course, because if the intensity is too low, below a certain level, even the moths, which have very specialized detectors, cannot perceive the presence of the odor. The quality is important, like I told you before. Moths use a blend of different odors, and the various ratios of these components of the blend matters a lot, as we will also see at the end of this part. And then finally, what matters a lot is also the structure of the signal. So as you see here in the example, if you receive a message which is mangled like that, it's very hard to reconstruct the original message. But if you know that the way that these letters have been erased follows a complex but a learnable pattern, then you might be able to decode even messages that are so noisy and corrupted. Okay, so first, intensity. How is it even possible that that a moth can detect a signal, such a faint signal, from hundreds of meters away from the source. Clearly, odors get dispersed in the atmosphere. So the first thing that that is fascinating, of course, has been a big riddle for a long time. How can moths possibly react to the signals? And actually, it turns out that moths' antennae are actually in an exquisite sensor device for odors. Experiments in the laboratory have shown that as little as 10 molecules that hit this antenna in 1 second are able to elicit a change in the cardiac response, in the cardiac beating of the moth. So these are really sensors which push their ability to the physical limit of detection. Okay, less than this, that probably there's nothing to sense, right? We're very close to the physical limits. How do they work? They basically are essentially some molecular sieves. Okay, so every antenna has several hairs, several branches, and each branch has several hair. You see, in the tens of thousands of them, and every hair is actually a detector of flux of molecules. So molecules come in, enter this part where it's some lymph, and then eventually they make contact with the olfactory sensory neurons, which then send out the message to the peripheral nervous system and so on. Of course, what happens from here onwards is extremely interesting and fascinating, and you will hear about that, I hope, very soon. So this is basically the way by which even a single molecule hitting an antenna is able to elicit some response through a series of amplifications and discrimination processes. But the part that we will be mostly focusing upon now is about the structure of the signal. Okay, so this is you can see here it's a snapshot of a concentration of a dye. Okay, so or a smoke. You can think of this as smoke in the air. It's not a pheromone, but it's sort of a proxy for what a pheromone does. This is a picture taken in a laboratory, so in a wind tunnel. It's not even close to what happens in the large-scale atmosphere, but it's somehow already gives us an idea, a visual idea of what happens when I say that the signal is broken. So at the source, the signal is emitted here. There is some wind there, and as a consequence of the fact that this wind is turbulent, you see that the original flow of odor, if you wish, of smoke here gets very rapidly contorted and distorted. So if you are just 1 meter down of the source, what you will see here in time. So if you put your sensor somewhere here in the middle and look at what happens in concentration, if your sensor measures this in time, you will see that for very long periods of time there is basically no signal at all, and only from time to time you have these excursions, which we can call a whiff of odor, and these whiffs are separated by blanks. And sometimes you can also have clumps of these whiffs altogether. And the key point is that all these whiffs and the time spacing between them are not, strictly speaking, random. Okay, so there is this process by which turbulence carries the signal has internal correlation, is a complex, non-linear process, which, however, has some reproducibility. Okay, so the statistics of which with which these whiffs come one after the other, or how many of them can stay close in a clump, all these things are in principle predictable, knowing how the dynamics of how fluid dynamics works. In the real situation in the field, what you see, if you put a sensor hundreds of meters away from the source, so you enlarge the scale of the system to hundreds of meters, what you will see is that some of these whiffs are actually as short as few milliseconds. So these are very, very short signals. And the time that can pass between one clump of WIFs and another could be even several minutes. So the signal in the wild is extremely, extremely sparse and extremely intermittent, which makes the task of recovering the source extremely difficult, as you can imagine. So you cannot rely on any notion of saying, okay, the concentration of odor is higher when I'm close to the source and is lower when I'm away from the source, because you have to face the fact that there can be minutes in which you don't get any signal at all. So you cannot estimate anything in principle if you don't have any signal at all. At least you cannot estimate a gradient. We will see how actually you can integrate this absence of signal also as a way to process information, provided you have some prior knowledge of how the system works. Okay. So this is just to highlight the difficulty of the process. Yes, Vijay?
There are a couple of questions, Antonio. So in the previous slide, what is the blue line? This is some average concentration.
Okay, yes, thanks for asking. Sorry, I'm going the other way. Yeah, right. So in this, this blue line here is the average concentration given that the signal is non-zero. Okay, so here the signal is virtually zero. So if you just consider all the excursion and take the average, then this would be your capital C. And here I'm depicting what happens, assuming that the threshold for the sensor is placed here. Then if this is the case, then there will be encounters of odors only when the red triangles are marked.
There are also a couple of questions, more general ones, earlier part. People wonder if the odor of the female of one species can attract males of another species. Okay.
Yeah, I will come to that at the end of this first part, but the short answer is no, and it's quite important that this is not the case, because you don't want to go a very, very long way and undergo scrambling competition with other males and then eventually find out that this is not and nothing's going to happen basically for your survival.
Sure. Another question is that suppose there are two females and you're getting signals from both of them. How does the male decide which one to go?
Okay, yeah. So thank you for this question. This helps me clarify some aspects. So this approach and scrambling rush towards the female is only the first part of the mating process. When males come close enough to the female, then of course they started to see her and decide what to do. And then it also starts a complex mating ritual. There's all sorts of dances and approaches, etc., which might or might not lead to mating or not. So this is just one part of a much longer process, which also involves several other steps.
There was a hand up, now it's down. I'm guessing there's no other questions.
Okay. Maybe when I'm done with this first part, I can also collect more questions. Okay, so all this was to make sense that for you to appreciate that the difficulty of the task that insects have to face for mating. And this is a quantitative expression of the richness of the complexity of the signal that you experience with the sensor in the atmosphere when you're hundreds of meters away from the source. You see the duration, like I told you, the duration of a whiff can go from milliseconds to tens of seconds. And the time between two whiffs, again, has this very broad and power law distributed laws, which means that there is a very complex and rich spectrum of time scales and signals which is generated by turbulence. And of course, since this is everything as the opposite as a featureless signal, there is information to be extracted from this series of signals. Okay, so if you work out the theory of how transport of odors happens in the atmosphere, you actually can come up with estimates. And if you put the actual numbers for the size of the source. There's the size of a moth, typical wind, typical fluctuations of the wind, typical thresholds for detection, which are obtained from experiments by ethologists and neurophysiology, the typical concentration at the source. And then if you put all them together, you come up with estimates of the order 1,000 meters, which is essentially what you expect for the highest possible range of attraction for a female. And so you can go on with this exercise, and basically the point is that we by now have a quite reasonable understanding of quantitative, semi-quantitative understanding of how the various timescales and properties of olfactory communication turn out and give quantitative estimates of that are useful in understanding when the process can and cannot happen. Okay, so I will skip very quickly through this because what I wanted to show you is a few experiments that have been done in the past. As you see, people have been trying to capture this phenomenon since 1925, but more quantitatively in the last 50 years. So one of the first and most useful devices is to use a wind tunnel for observing moths. Okay, so here there is a camera. You put inside a moth. There is wind flowing here, and then there is maybe a cloth which is imbibed with pheromones, which therefore gives the signal for the moth to fly against the wind and reach the location of the origin of the odor. You will notice, so here in the figure below, you can see that this is an actual picture of this device, and this is obtained through some smoke. So you can see, even though this is not very similar to what happens in the wild, because the signal is basically everywhere there. You never have very long periods of time where you don't get anything unless you're very much away from the center of this so-called plume. But you see that the broken character of the concentration is already visible at this in this very simplified setting. You will notice these stripes here at the bottom. So these stripes are important for navigation for moths and flying insects because they provide the visual cues by which they can appreciate the progress that they make with respect to the ground. Okay, so what's the problem here? So moths have to fly in a medium which moves itself. Okay, because they have to fly against the wind. So going upwind is a good decision because if you perceive some odor, it's very likely that it's coming upwind. Okay, the probability that it's coming from your back, it's relatively low, exponentially low actually, if you make it quantitative. So it's a good idea to search upwind, but when you face the problem of flying upwind, so you can imagine this with an experience which is probably more understandable to humans, is that imagine that you have to swim in one of those pools where there is a current flowing towards you, okay? And you're trying to keep up with the current, but unless you have some visual cue at the bottom of the pool, you cannot really understand whether you're making progress towards one side of the pool or the other. The only thing that you can sense the presence of the current against you, which is what is called anemotaxis, but you have to combine it with the optical visual feedback in a process which is called optomotor anemotaxis. And this is what provides the moth the ability of deciding to move in a certain direction with respect to the ground, notwithstanding the fact that there is some wind pushing against them. But we are not interested in the interplay between visual, mechanical, and motor stimuli that give rise to motor decisions. We are interested in what happens to the odor. And the first thing that appeared quite clearly in this kind of experiments when you use odors is that it's very important the structure of the signal that is received by the moths. Okay, so in the first experiment, suppose you just start exposing moths to a constant concentration of odor. So at time 0, you put your cloth or you open the box where the odor is, and then the odor starts flowing, but it's uniform. Okay, there is no pattern whatsoever. This is not what happens in nature, but you just try with this. There's a cloud of odor going towards the moth. Then when the moth starts perceiving the presence of the odor, it starts moving around. It can fly, it can flap its wings, it reacts by some specified definition of the assay. But after a few seconds, it subsides, it just stops. Okay? So it doesn't react to this uniform cloud of odor. But on the other hand, if you subject the same male moth to a sequence of on and off odors, so clean air odor at different frequencies, then this tends to elicit a more continuous fly upwind. Okay? So the frequency at which the signal is detected appears to play a very important role. And this is well made clear by the following experiment. So this is exactly the experiment I'm describing. Here there is this is, if you see, it's a section of the wind channel seen from the top. The wind is flowing from the top to the bottom here. This is the span of the wind channel, and this is the longitudinal direction. The odor is coming from here. And here the odor is administered in a series of pulses. Okay, so odor, no odor, odor, no odor. And what changes here is the frequency at which it is the alternation of signal comes. So for instance, for the top, the signal is given in a sort of adagio tempo, that is means. Whereas if you look at what happens at the bottom, this is a very fast pulse. Okay, and you can clearly see the difference in typical behaviors. Okay, when the signal is not very frequent, there is this alternation of sideways oscillations with progress upwards, but a slower one. Whereas when the signal is very fast, there is a tendency to do more straight trajectories upward. So in these experiments, it was also possible by administering single pulses to observe this change of behavior from sideways oscillations to upward flight. So for instance, these are 4 selected examples from the same paper. So the trajectory starts here, and the insect here is moving sideways, crosswind. Then with slight deviations, then it makes contact with the pulse here. Okay, so here I should stress that this is a very difficult experiment in the sense that at any time the odor is not seen, so you have to deduce the presence of the contact by other means. So it's quite delicate, but nevertheless, this is the bona fide point of contact with the pulse, and then as a reaction with some delay, then there is this part of upward wind flight, and then again it resumes a more oscillating pattern. Okay, so this seems to be some sort of hardwired response of behavior to single odor encounters, which was emerging at the time as a paradigm of stereotyped response to odor encounters. And of course, this is modulated by the frequency, because if you have many encounters, basically you are always locked in this flight, upwind flight. Whereas if there is more sparse, then there is time to make oscillations. Okay, so in the same spirit, very ingenious experiments have been done. For instance, in this experiment, a third antenna, so most typically have 2 antennae. Then here experiments have taken a third antenna from another moth, and they have implanted on the thorax of the moth and have measured potentials across this third antenna, so using it as a sensor. So basically, the moth is equipped with a small backpack with an electron-antenogram, and then the signal are recorded during flight. And after that, there is an alignment between the trajectory that is observed and the responses measured at the level of the antenna during flight. Okay, so this is quite amazing. And since then, I must tell you that there's been a lot of effort in this direction, including cyborgs that mix real antennae with electrical engineering or robots, etc. So it's a field in itself. Here you can see the kind of experiments that you can do with this site. This is a trajectory, and these are the positions where the potential drops, which are considered to be associated with encounters with the odor. Okay, even though of course the association is a bit indirect. Okay. How much time do I have on my first part? Maybe just 5 minutes. Okay, so I can go very quickly through this. Unfortunately, there are limits to experimental limits to the kind of signals that you can subject an insect to in the environment. Just because the release of the odor just takes some time, there are latency periods, etc. So one interesting attempt at investigating the speed of the response of moths to different time odor patterns was obtained by using optogenetics. So basically, receptors for odors receptors for light were implanted into the antennae in such a way that light now replaces the stimulus instead of odors. So the response from the receptor downwards towards the central nervous system is the same, but the receptor now responds to a specific light of a specific wavelength. So using this trick of optogenetics, it was possible to actually to see that pulses of light as short as a few milliseconds already elicit a response in moths. Okay, and when they are longer, like 10 milliseconds, then they are essentially saturating the response from 10 milliseconds on, which is an extremely short time for the response of the neural system. And very interesting, it's exactly in the ballpark of the shortest pulses that can be observed in the atmosphere. And also, it's possible to show with by the same technique that there are synergetic effects. So if two very short pulses are presented one after the other in a very close times, then they can elicit a response much larger than when they are far apart. So very fast signals and their spacing matter a lot in eliciting the response of moths, as we come to understand. Of course, all of this was inside the wind tunnel. But what happens when you go into the wild? The physics of the signal changes. Qualitatively, it's also interspersed, but the quantitative aspects are very important. There are long periods, up to minutes, in which the moth cannot see the signal. So it's very difficult to follow tracks of moths in the wild. One attempt was done now almost 30 years ago. In these experiments, moths were painted with a pink color on their back, and then observed from a tower which was 30 meters high over a domain which was about 30 meters long. Okay, again, this is not very much short of having the hundreds of meters that we would like, but that's basically and still is essentially what the best that has been possible to do in the wild in order to track the trajectories of moths in a reliable way. So moths then were videotaped, and at the same time there was an attempt to track the local direction of the wind by seeding soap bubbles and seeing where they were going. Okay, so everything was very a lot of craftsmanship in doing these experiments, and the kind of trajectories that emerge here on a much larger scale than the wind tunnel. So the wind tunnel would be just like a box of this size. Are quite interesting. Quite interesting because you see that there are these very long trajectories across the field, and then relatively long periods of stretches. And from this observation, it's so these are very sparse, so it's not possible to do any kind of statistical analysis of these trajectories. But it also points to the fact that the process might be way more complex than just this simple sequence of stereotyped behaviors that is just like move sideways and go up with when you detect. So this is basically one of the outstanding question, whether this very simple model of response to the pheromone plume, which is a sequence of surges upwind when there is detection and casting behavior when there is none, is an appropriate description of what happens, whether there are memory effects. So I cannot go into details into this, but this is just to sort of to give you a quick idea of the kind of proposed reactive strategies and what actually could be there more, which involves also memory, short-term or maybe long-term memory in this process. To finish up this first part, I would just quickly move to another kind of experiment, which is sort of trying to bridge the gap between what was possible to do with moths in a wind tunnel and what we would hope for in a different setting. So this is an experiment in which there are walking flies. So these flies don't fly, but just walk in this arena. Odor is coming from the left. There is a jet, and there is a perturbation system here of lateral jets, which tries to mimic as much as possible the statistic of encounters that an insect would perceive at a much larger distance from the source than the one that is present here. So it's a sort of way of generating a synthetic odor signal which mimics more the one in natural conditions. So here you see that the kind of signals that is observed by the flies. The interesting thing of this work was that it's one of the few situations in which you can visualize at the same time the position of the insect, its movement, so you can track its movement, and at the same time you see the odor, which in this case was smoke. So quite serendipitously, the authors discovered that moths were attracted by smoke, and so they used this kind of smoke to track the presence of the odor and to follow the trajectories. And as you can imagine, this gives rise to a lot of high quality data in which you can basically map the presence of the signal into the decisions that are made, and then you can try to fit behavioral models. And one of the emerging results from this kind of work is that walking flies typically switch between two behaviors. That is, either they stop or they walk. And when they walk, they can perform turns either upwind or downwind. And the frequencies by which they switch from one of these states of behavior to another is random, but it's modulated and it's biased by the frequency of previous encounters. Okay, so this, even though the system is different, the biology is different, this also points to some common idea that the sequence of detections that the moth or the fly has made in the recent past provide a very useful input for the processing system to decide what kind of navigation decisions to make in order to approach the source. So this kind of approach could probably represent a template for what one would like to have in an experiment. So careful control of a dynamically changing environment, like the one that is produced by turbulent jet, and at the same time observing stimuli and behavior simultaneously. Okay, so with that, maybe I just I will just answer very quickly to the question about blends. So this is a very interesting and telling example. So here you see three pictures of corn borers, which are pests that as you can imagine, are very, very harmful for corn crops. So there are two species here. One species is the European corn borer, which comes into two strains, strain E and strain Z. And then there is another species, which is the Asian corn borer, which is an entirely different species. So males of European corn borer don't mate with the Asian corn borers and vice versa. So the difference between these two strains is that both of them respond to a different mixture of two pheromones, which are different only by the spatial positioning of the molecules. So this is the same molecule, 11-14 acetate, only two stereoisomers. So they are the atoms, the molecules, the atoms in this molecule are arranged differently in space. So this strain here only responds to this E form of the pheromone, and the Z strain here only responds to the Z form. However, the Asian corn borer, which is a different species, responds to a mixture which has almost equal proportions of these two stereoisomers. All right. So here you see another difficulty in the problem of mating, in the sense that suppose that this is a male of the Fumacalis, which responds to the 50-50% mixture. And then far away there are two strains, two females, one for each strain. So this one emits only E's, this one emits only Z's. But what the Fumacalis sees, the male sees from a distance, is just a mixture, because far away from the source, then these two blends tend to be these two odors tend to mix each other and form a blend. So how can a proper decision be made? How can the insect decide that it's worth flying towards the females or not? Of course, this is one interesting question in itself, even though it's a bit contrived, because these two species don't overlap geographically, but you can imagine just the kind of question that you can extend to more realistic situations. The interesting thing is that the statistics of the odor concentration tells you that a solution is possible, in the sense that if you detect a signal above a certain concentration, then it's quite likely that you will find either only red or only blue. So if you move around and eventually hit a patch of odor which is mostly red, then you can have the certainty that there is a red emitting female at the other end of the line, and this makes it worse. It makes it worth to fly. But this depends crucially on the parameters of the situation, the sensitivity, etc. Okay, so with that, I think I'm done with the first part. So I just touched upon many subjects, and but if you want to look deeper into this, so this is a very good reading. It's a book which discusses olfactory communication in general, so including many other things that are not covered by large distance olfactory search. And then this review, which is relatively recent, is also an exceptionally good entry point to see many different aspects of olfactory communications that I just very, very briefly skimmed upon. And I encourage you to read and to delve deeper and to find all the proper referencing in this paper. So with that, I think I can stop for the first part and take questions, and then we can move to the second part.
A couple of questions, Antonio. So Shrivas asked if there are multiple moths releasing pheromones in the same area and different kinds of pheromones. What about the magnitude of the mixing? And is that a relevant factor in non-lab settings? And has this been measured? Okay, yeah. I'm not sure if this example is somewhat gave a meaningful answer to that. If not, please expand. So if I understand correctly, the question was about having several different mixture and now they are mixed by the environment. So that's certainly an outstanding question. And but if I'm not getting the question properly, please write again. Shivas, you want to unmute and ask? Shivas? We can't hear him. Okay, I can answer to the question I'm reading now.
I'm not sure this has been
Hello,
measured.
can you hear me now?
Yeah, go ahead.
Hello.
Yeah, go ahead. Shivas, maybe we can come back.
Am I audible?
Yeah,
It
you are audible.
tells me I'm audible.
You are audible. All right, so we can come back to him. Pooja has a question about the particular length of the patterns that you would provide at the bottom of the wind tunnels. Does that provide the moths a sense of movement?
Yeah. There is a proper way of choosing it, because as you can imagine, it's a combination of the ground speed of the moth while in flight, and its ability to measure the changes in patterns with the frequency. If it's too sparse, it's not good, and if it's too small, of course, it just gets blurred. So there is a sweet spot. Off the top of my head, I cannot say if it's extremely sharp, or there is some leeway to provide as a sensory. But I know that there have been other experiments which provide more naturalistic patterns, so virtual arenas in which there is actually a landscape flowing, and this also produces good results in automata and hemotaxis.
Right. Sanjay Sane's lab works on that. I don't know if he's here. So Priya has a question about why is there a frequency dependent behavioral difference, the directional movement?
Why is there a frequency dependent behavior? Why? Yeah, why is always a very good question. Yeah, so one possible way to address this question is that if you encounter many odors, if you make many odor encounters in a short time, this is a signal which contains specific information about the location of the source, and which distinguishes this location from others. Okay, so typically, if you make many encounters, then it's very likely that you're sitting along the line that connects the location of the source along the wing. So the probability of seeing 3 detections in 1 second is much higher along the line than it is, say, 1 meter away. So this is valuable positional information. So we can only postulate that this kind of encounters make the moth take the decision of surging upwind more with a better frequency. Of course, everything is uncertain in statistics, so you don't commit 100% to this, but you can use this signal with more confidence. We will try to make this mathematical tomorrow.
Fernando has a question. I don't know if he's still in the meeting. He has a question about, is there other experiments where animal olfaction is more specific than human chemical methods?
I advise you to ask this question to my colleagues, and probably already Dima next talk will be able to answer much better than I do on this.
Okay, so there are 2 raised hands. So Pramod, can you go ahead?
Yeah, so my question is that from what I understood, there are 2 types of information the moth is receiving. One is the tempo of the signal, and the other is the wind direction. And I suppose these insects also have sensors to measure wind direction. So is the moth using both, or is it only using the tempo to do the search?
No, it is actually using both. To what degree it uses the wind direction, whether it's very refined or just coarse information, we don't really know. But it's crucial to know what is the average wind direction. Okay, so this also requires a little bit of integration, because as it flies, it has to compute the perturbation induced to the wind. So it requires some coarse graining in time to understand what the mean wind direction is, which is a relevant information for locating the source. Whether it uses finer structures, that is other fluctuations in the wind or not, it's not obvious.
So are there any experiments which try to decouple these two, either by having some clever experimental scheme or by deactivating the sensors which sense the order versus the wind direction?
Okay, yeah. I think that a common practice is to perform experiments with wind but without odors and use them as control. So in order to tell the difference. So this is typically done.
Thank you very much.
Sure.
Sanket has a question.
Yes. Hi, everyone. So my question is basically on like two slides. For one of the slides, we saw that when the duration of the pulse is like it is continuous. So then the mod does not move and like it moves partially and comes back to its original location. On the contrary, when we are there's another slide and which shows that as the duration of the pulse increases, more mods move towards the stimulus. So I want to understand that when we are kind of increasing the duration of the pulse towards infinity, the mod tends to come back to its location and not move towards the stimulus. So aren't these two contradicting, like in a sense?
So let me try to go back to the slides to see whether which one you're referring to. So are you talking about this?
Yeah, this one.
Yeah. So okay, let me give you some more details. So in this case, the response of the moths for this particular experiments was wing flapping. Okay, so these moths are not taking flight yet. So they are there. They are exposed to this brief light, which simulates the presence of odor, and they start moving their wings, and so that's the response that they have, but they don't fly. And I don't remember off the top of my head, but it could as well be possible that if the duration is too long, then this response subsides again. So if you extend this to second, maybe it just subsides. I don't know, and I don't remember if there are data for this in this experiment, but I don't see any conflict because the time scales for habituation are typically longer than this subsecond scales.
Yes. Thank you.
Okay, you can go ahead.
Okay, so I will have to share another set of slides for the second part. Okay. Okay, I have to go out here. Not just this. Share. And that. Okay. So I think I have more or less half an hour, so I will be slightly quicker. But I would like to tell you about a totally different biological system and actually a totally different task, which is, however, very interesting and in which olfaction, surprisingly enough, plays an important role. So this is about homing pigeons. So this is a very common rock dove, which you might have encountered many times in your life. And the important thing for us is that these doves are capable of an incredibly complex task, because if you raise the doves in a loft for a few months, okay, and you allow them to fly a little bit around your loft, maybe takes short flights of 100 meters, 200 meters, they're still learning to fly. And then when they are, say, 4 to 6 months old, which is basically the first, when they are start being adults, you take these pigeons and you drive them hundreds of kilometers away from their loft to a place that they have never seen before. And then you release them, and most of the time they get back home during the day. So these are totally inexperienced pigeons put in a totally unknown environment, which nevertheless managed to find their way back home. The big problem with pigeons, which I have to tell forehand, is not their ability of being able to go back home, but it's about their motivation. Pigeons are very social animals. So what often happens in these experiments is that when you release them far away, when they start to get back into a zone, they encounter other lofts, they encounter other communities of pigeons, and they say, okay, these guys are way cooler than the old guys at home, and why don't I stop by and stay here? And but this makes a very big difference with respect to moths, because moths are extremely motivated to do their task, whereas pigeons might be sometimes lazy. But nevertheless, over the years, there has been an enormous amount of experimental evidence that this kind of homing behavior is reproducible and has very specific quantitative characteristics. So birds are able of navigation on planetary scales, right? So they can fly across the planet. So it's important to distinguish different ranges of navigation, because different ranges have different specific cues which are relevant. So there is a long distance phase which takes place on thousands of kilometers, which is the one which is relevant for migration and for long distance travel. Then there is a short range navigation, which is mostly based on familiar places. So there is probably a topographic map that the bird has about its surroundings, and it uses it to navigate, to go from the lake to the perch, etc. And then there is this intermediate range of scales, which is hundreds of kilometers, which is the one which is relevant for homing. Okay. So, so in this picture, I'm giving you one example of the experimental evidence of how pigeons home. So remember, these are inexperienced pigeons. First time that they go away from the loft for more than 1 kilometer. Here there are two sets of pigeons. One which is whose home loft is here in the north of Germany, Wilhelmshaven here, and the other one is here in I guess Gießen. And they are labeled by white circles and black circles. So pigeons from here are taken either here or here. These are two different release sites for the black pigeons. And the white pigeons are taken from here, either to here or in the north of Germany, close to the border with Denmark. And then they are released. And here, all these arrows that you can see in this picture, these arrows that you can see in this picture, which are also reproduced here, give information which is called the vanishing bearing of the birds. That is, when they are released, the experimentalists look at the direction that is taken by the bird. So the bird stays around for a few minutes, gets acclimated at the place, and then starts flying away. And then they record the direction that they have last seen them before they go out of sight. And as you can see, for the birds which were grown here and released here, they all consistently tend to point south, with some fluctuations, with some deviations. And in this case, a large fraction of them was eventually recovered at the breeding site. And from the same site, those that were left in this release site at the south pointed northward. And same here, okay? So they seem to have already when they start their journey back, already an approximate idea of which is the direction to go. And here on the right, you see some of the first GPS tracks of these pigeons, which were, this was their home. They were carried by car to the release site and then left. And then you see several of these pigeons taking different routes, the routes that take them back to their home loft. So of course, this is particularly striking, and the big question is how can this possibly work? And so this is a navigation task. Okay, so one has to find a route towards a determined location. And as a general strategy for dealing with these navigation problems, one could rely on two tools. One is to have some kind of map on the environment. And the second one is to have a compass. Okay, so let's start with the compass, which is probably the simplest thing to imagine, to realize how it works, and then we will move to the map, which of course already looks quite kind of a challenge, because it's impossible that this unexperienced bird taken to unfamiliar places have a topographical map of the route that they have to take. So it must be something else. So the orientation information, well, the basic recognition is that if you have a clock and you can see the position of the sun, then this can work as a compass. Because by matching the time of the day and the position of the sun, you basically know where the direction of the south, for instance. Okay, so do pigeons use this sun as in the clock as a compass? So for the clock, they probably must rely on their circadian rhythm as a clock, as an internal clock. And then they have to see the position of the sun and probably then compute the direction in which they want to head. So in order to test this hypothesis of the sun compass, experimenters have done an extremely smart experiment, in the sense that they looked at the behavior of control birds and compared it with birds which have had their circadian clock shifted by 6 hours. So birds are grown in a closed environment with artificial lights, and these artificial lights follow a cycle which is shifted by 6 hours with respect to the natural light cycle. And if the hypothesis is correct, when the birds that have been shifted go out, rather than pointing in the direction of the home, which is what the control birds do, they should point in a direction which is shifted by an angle which corresponds to the fraction of the day that is the delay that has been imposed to their cycle. And this is exactly what is seen here in this experiment. So these black dots are the vanishing bearing directions of the birds when they are released, and you see that they tend to point towards the direction of home. Whereas the birds which have been time shifted tend to present a deviation of an angle which exactly corresponds to the shift in time that they have experienced. Okay, so this is a very solid evidence in favor that supports the idea that the sun and the internal clock work together to provide a compass. But what happens if the sky is overcast, which is, of course, an opportunity that can happen? Then there must be some backup compass mechanism. And the fact that this compass mechanism, alternative or secondary compass mechanism is present, is seen in these experiments. So if the day is sunny, the control birds move in the right direction. Okay, so these are the directions taken by the control birds, and this dotted line is the actual direction of the home. The shifted birds, they move at another angle. Okay, and they are also much more scattered. But if the sky is overcast, the shifted birds again point better in the direction of the home. So this means that there is some other backup mechanism that says, okay, if the sun is not present, I discard all the information about the circadian cycle and I resort to this other mechanism, and this one works in place of that as a second mechanism. And what this second mechanism is, well, it turns out that it is the magnetic field of the Earth. So this was also proven in different ways. And one of the ways that it was sampled is that by putting magnetic coils close to the head of the pigeons with a magnetic field which is of intensity comparable to the Earth's magnetic field, and then deciding whether to make it point, say, north or south, and seeing the responses of the birds with magnetic coils in presence of these currents. And what this experiment shows is that in the presence of sun, the magnetic coil is basically ignored. So the sun works as a primary compass, But in absence of the sun, then the orientation of the magnetic coil starts to matter, and the decision whether to go north or south depends on the orientation of the magnetic field imposed. Okay. So summarizing, these are just sample experiments, but there is an overall coherent picture that says that the compass problem for pigeons is basically made of 2 steps. So if there is a sun, use the circadian cycle in combination with the position of the sun to decide where is south, where is north, so how to orient yourself. And when the sun, when the sky is overcast, then resort to this magnetic field compass. Okay, so this is the part which has, of course, as you can imagine, nothing to do with odors or anything else. But the problem remains: if you have a compass, but you don't have a map, you can possibly find your way back home. So what is the map? What is the map? So there were several hypotheses put forward, but significant progress was made sometime in early 70s, when essentially by trial and error, a group of ornithologists from Pisa discovered that if they severed the olfactory nerves of the pigeons, these pigeons were strongly impaired in the process of going back home. So they had 2 sets of pigeons. One of them had surgery and removal or cutting of the olfactory nerve, and the other one, the control set, had a sham operation in which they were subject to surgery, but without cutting the olfactory nerve. And then the performances in homing of these 2 groups of birds were compared, and the ones which had the sham operation performed very well, just as ordinary birds, without any operation at all, whereas the ones that had their olfactory nerve cut were not able to go home. So hence the hypothesis, olfaction must possibly have something to do with this orientation, but how could this possibly work? So after several experiments, some of them I will tell you about because they are quite intriguing and actually fascinating by themselves. The emerging picture is the following one: is that there is actually an olfactory map, which is built as follows. So during the period at home, so from birth to this 5 or 6 months of age, the pigeons are in their loft, and they are naturally exposed to winds coming from different directions. And winds coming from different directions typically have some distinctive odors, are associated with some distinctive odors more than others. So for instance, if the pigeons are grown up in a position where there is, let's say, the sea is 50 kilometers to the west, and then there are forests to the north, and then there is a big city to the south, all these odors of the city, odors of the sea, odors of the forest, they come towards the loft, depending on the direction of the wind. So the first assumption is that during the development, the bird learns to associate wind directions with typical odors. In order to navigate in a 2-dimensional environment, 2 odors would be enough, but very likely they use maybe 10 or 20 different odors and different directions in order to construct these associative maps between wind directions and odors and prevalent odors. And then how do they use it? When they are taken away from home up to a certain release site, they start sniffing the air, and they realize, okay, so here there is a majority of blue odors with some yellow odor and some red odor. So this means that since these odors are blue, are in excess with respect to the ones that I measured when I was at home, and they come from that particular direction, I must move in the same direction from which they went home, right? So I have to follow the leading odor. If there are many of them, there must be some rule to combine this multiple information into one heading direction. So this is a hypothesis, olfactory map hypothesis. So how can this hypothesis be tested? Okay, it seems quite a very difficult task, but there are several pieces of evidence that go into this direction over the years, and I will review some of them in the following. So the first experiment is extremely neat. Pigeons are bred in a channel in which artificial winds are produced, and these artificial winds are associated with specific artificial odors. So, for instance, there are some fans that send wind from the north direction, and there is some turpentine, which is a very pungent odor associated with these winds. And when these winds are coming from the south, they are associated with olive oil. So there is olive oil present there, which is basically blown onto the pigeons. And these triangle pigeons are trained to make this association. Okay, so that's the hypothesis. And then there are these diamond pigeons, which grow in the same place nearby, but they are exposed to the complementary pattern. So olive oil is coming from north. Then when they are 6 months old, they are taken away again, here 20 kilometers away from their home, and they are exposed at the site to either olive oil or turpentine. So if they are exposed to olive oil and they are the pigeons who are triangles, then they believe that they are finding themselves south of their home, because that's where the wind is coming from. And so they head north. And oppositely, if the odor is coming for the diamond pigeons, etc. Okay, so these consistent patterns of orientations when released is a circumstantial evidence in favor of the olfactory hypothesis, and this is one. Other very, very smart experiments. Pigeons are grown in lofts in which there are a series of deflectors which change the direction of the wind. So a bird which lives here gets the wind coming from south, arrives through a deflector, and so it makes it appear as if it's coming from east. And here in the other way around. So these are two ways to deflect by 90 degrees the original direction of the wind into the one perceived by the pigeons. So under this hypothesis, the responses of the pigeon when released should be shifted accordingly. And this is roughly what is observed here. The control pigeons head towards home, whereas the right deflected pigeons turn towards left, and the left pigeons turn towards right, which is again consistent with the idea of this learning process going on during development. Other experiments. In this experiment, pigeons, which are bred here, are taken to the release site into two different groups. There are two different groups of pigeons. One group is taken by making a long detour to the north, and another one is by taking a long detour to the south. During all this period, the pigeons are exposed to external odors, so they can sense the odor of the air they are flying into. So according to the olfactory map hypothesis, the pigeons that travel to the north should sense more north-coming odors, and therefore their direction when they go back home should be shifted accordingly. And the opposite should happen for the birds that are taken along the counterclockwise route, which stays more to the south with respect to the other one. And in fact, when you look at the bearing directions of the birds when released, the two populations are clearly separated in the direction that you would expect according to this hypothesis. And then there is a variant, which is an even more elaborate variant of this experiment, in which is called the false release site experiment. So pigeons are grown here at this home, and then they are carried to the release site placed here in R. But they are carried to a very specific road, okay, which is given here in this dashed line. So one group of pigeons goes to the south, And during this travel, they are not exposed to odors. So the box in which they travel has filters, so there's no perception of the external odor as they travel until here. So when this is what is called the false release site. So here they are exposed to the air, so they can sense what the air is here, the concentrations. Then they are closed again in a sealed box, no odor, well, not sealed, I mean a filtered box, and they travel to the release site. When they're left to the release site, they are anesthetized so that for 1 hour or two, they cannot sense the outside odors. So the idea of the olfactory hypothesis is that when they are released, they would react depending on the last input of odors that they had. So pigeons that have sensed the odor here should go north, and pigeons which were taken to the north false release site and then taken here should go south. And that's what they do for a time of about 1 to 2 hours. Then when the anesthetic ceases, then they start correcting their way, and then they eventually find their way back home through a convoluted road. Okay, so this is just a few handpicked experiments, but there's a wealth of them that has created a general consensus on the fact that this olfactory map hypothesis could be exactly the way that that pigeons work on this. And but the big point was whether are these long-range gradients of concentration, do they actually exist in nature? So Hans Warraff at some point decided to make an extensive investigation. So he went around for several years in a row, moving around in an area of hundreds of kilometers and taking samples of air in 100 different locations in an area of 300 kilometers of diameter, and then doing gas chromatography to detect the presence of several chemicals. And then by this analysis, he was able to sort of conclude that in fact there were a relatively stable average distribution of concentrations, which tend to be, for instance, benzene, which is a typical byproduct of human activity, tends to be higher in the upper part of this map, whereas isoprene, which is typically associated to woods, is more intense on this upper corner. So this was sort of proof that these large-scale concentration gradients actually exist. But of course, this can only exist on average, because the atmosphere, once more, is turbulent. So if you take, so this is the result of a numerical simulation, but this is just to show you what happens when you have a gradient of odor. So say that this red odor is a gradient from north to south, and the green odor is a gradient from east to west. But these are the actual concentration that you see at any given time as the system evolves. So it's far from unlikely that you will experience an excess of red odor when you're to the west. So how should you interpret that? How is it possible to navigate again in this very non-linearly distorted environment in order to be able to reach home? Okay, so these are again kind of questions that are different in nature with respect to the problem of olfactory research, but fall into the more general problem of how to interpret and how to make decision in presence of a very complex landscape of signals such as the one given by odors in the atmosphere. Okay, so I'm done with the second part, just in time. Again, I encourage you to read more about this. This is a very nice book by Hans Warraff, which has been together with Papi, one of the main advocates of this olfactory navigation hypothesis. And this is also summarized very, very well in a shorter read, which is the review by Anna Gagliardo. Before we move to the question, very quickly, an outlook on tomorrow. So summary for today: olfactory research and navigation are very difficult task because they are long-term. So there is a sequence of actions that has to be taken in order to achieve the goal. And the environment is dynamic and extremely uncertain. So these are very typical conditions for animals. Besides the olfactory navigation, this is sort of a common situation, common challenge. It is also the same value when you think about artificial agents like robots. So the general question that I will try to address next time is whether there is a mathematical language that can describe these kinds of tasks in a way that is independent as much as possible from the specific details of the problem at hand. It's systematic and it's predictive in the sense that it can provide answers to the question, why is the agent reacting in this way? So is it trying to optimize this kind of quantity? Okay, all this mathematical language actually goes under the name of decision process theory and reinforcement learning, and I will try to give you a short presentation, short introduction to the subject in the lecture tomorrow. With that, I think I'm done. Thank you.
Thank you, Antonio. Wonderful talk. And a couple of questions. So one from Shashank, he wonders why is there a reason, if there's any, the sun to be the primary compass and the Earth's magnetic field as a secondary one? Again, a why question.
It's an excellent question. In fact, I don't know the answer. That's my first answer to the question. There are several mechanisms which are more relevant depending on the scale. For instance, on larger scale, tens of kilometers, then there are celestial cues, which also can be used. The magnetic compass is more important. So I don't know exactly why in the homing process, maybe the answer is in the book, which I have read, but apparently not so carefully enough to be able to answer to your question. But yeah, it's an excellent question.
I had one question regarding the sensing the magnetic field. Is this the purported singlet triplet transition, which is thought to be the one behind how you sense the magnetic field?
So, sorry, say that again.
So, how do the birds sense the magnetic field? How do they know the direction? There's some sort of conjecture about a singlet triplet transition.
I'm not sure. I'm not sure about the actual mechanism by which they sense the magnetic field. Yeah.
So Priyant has a question. Priyant, can you go ahead and ask?
Yes, thanks, sir. Professor, I have a very curious question, which has been like disturbing me since you talked about it. So how good is the memory of the pigeons vis-a-vis their homing behavior? Okay, let's consider a hypothetical experiment where we release the pigeon from the same site multiple times. And in this case, would the pigeon follow the same route that it took initially, or does it take different routes to get back to its home? In this case, let's also assume that other parameters like the weather is considered to be the same for all attempts of this hypothetical experiment.
Yeah, sure. Thank you. That's a very good question. Yeah. So as a matter of fact, the pigeons change their behavior with experience. So these were unexperienced pigeons, first flight away from home. If you take the same pigeons and give them the same task repeatedly, they learn to do it much better and much more consistently. And they actually learn to use several landmarks. So they tend to fly following highways, for instance, or following valleys. So they learn, and it's actually a lifelong learning process. And as a matter of fact, there are homing pigeon races, which are quite quite a big deal, especially in certain countries, in which the pigeons are trained to go back home for 1,000 kilometers away from the home.
Wow.
And so they do this several times, and then these pigeons just the first one who gets there, and there's a as you can imagine, a lot of betting, and so these are particularly famous in China, and is also by these races that the researchers have discovered that anthropogenic signals are very important. So the presence of the city of Beijing was an invaluable cue for homing for all pigeons in this mechanism by combined olfactory and repeated training. So in a nutshell, the answer is that the memory, yes, it exists, and it's a process which by which you can actually train pigeons to be very effective in homing, and this is essentially why they were famous during the war for carrying messages, taking messages from the front line to the back of the troops.
Great, Professor. Thank you so much.
Any other questions? Okay, so let's thank Antonio for a very nice talk. Remember, you can also connect on Slack and put up more questions for him offline. I'll request Antonio to check it every now and then.
Sure.
Thanks, Antonio. We take a short break now, and we come back in 24 minutes, 7 p.m. Indian time for DMAS talk.